All right, so it's my pleasure now to introduce Emma Ponce. Emma is in her second year of her PhD here at Rasmus, and she'll be talking about a study looking at the effects of hypoxia, reduced oxygen levels on Acropora cervicornis.
Thank you very much. Let me go ahead and share my screen. All right, so thank you again for the introduction, Dr. Langdon, and thank you everyone for coming today to hear my talk. Today I'm going to be presenting some preliminary data aimed at determining the critical PO2 of Acropora cervicornis. So introducing my study subject, which is Acropora cervicornis. So ACERV is a beautiful scleractinian coral that has a symbiotic relationship with algal cells called zooxanthellae. So corals like cervicornis make up the most productive and biodiverse ecosystem on the planet. And they also provide crucial ecosystem services such as complex habitat formation, wave reduction, and pharmaceutical compounds. Acropora cervicornis was the predominant reef-building species in South Florida up until the 1980s, when there was unfortunately a massive white band disease outbreak that decimated about 80% of the original population. So these cervicornis populations continue to be threatened today by things like pollution, global warming, acidification, and disease. And I wanted to introduce another threat today that I think is becoming something that we need to be more aware of in terms of coral reef persistence in the future and in the face of climate change, and that would be hypoxic events. So these hypoxic events have been occurring more frequently worldwide. In Panama in 2016, we saw a mass die-off of a coral called Agaricia lamarcki. And you can see in these photos in the bottom that I'm showing down here, these panels A, B, and C, is the same colony of Agaricia lamarcki that was decimated over the course of several months. And similarly, in the Gulf of Mexico with the Flower Garden Banks, we saw mass coral mortality in several different species of coral. So in these photos over here, panel C and E, I'm showing on the top in panel C is a colony of Montasheria cavernosa, has a very ugly white looking kind of mat of necrotic tissue. And then underneath that in panel E is a colony of Orbicella faviolata that has a very distinct tissue mortality line. So in August of 2020, I'm sure most of you are probably familiar, we saw a mass fish die-off in Biscayne Bay that was attributed to reduced oxygen in the bay water. So it's of particular concern for us here in South Florida to understand how corals are going to cope with reduced oxygen conditions that we're expecting to see. So coral bleaching is a common response of corals to stressful conditions. I'm sure most of you are familiar with the process of coral bleaching, but it essentially is when the coral host expels its algal symbiont, leaving it kind of white and very vulnerable to other stressors and disease and kind of stuff like that. So in terms of the conditions that cause coral bleaching, they're generally things like chronic elevated temperature on the scale of degree heating weeks. So, you know, this kind of compounded amount of elevated stress. Additionally, low wind movement can create these kind of pockets of stagnant water with low flushing rates. And so that kind of stagnant water can become very stressful to corals and can lead to bleaching. So these two conditions that I just mentioned can cause coral bleaching, but they are also the same conditions that can cause a hypoxic event. So it's not totally unreasonable to think that these two things might be happening at the same time. And again, this is certainly not an exhaustive list of all the causes of coral bleaching and hypoxia, but the idea is that they can kind of happen simultaneously. So a little bit of background in terms of how corals utilize oxygen in their daily metabolism. Corals are known as oxygen conformers, which means that their oxygen consumption decreases when environmental oxygen decreases as well. So, and I'm including this equation as just a friendly reminder that the coral holobiont undergoes both photosynthesis and respiration, depending on those light conditions, right? So during the daytime, under high light conditions, its algal symbionts are photosynthesizing and providing oxygen to the coral host, whereas under nighttime or kind of cloudy or dark conditions, those coral host cells are respiring, and that process of respiration is oxygen dependent, whereas photosynthesis is not oxygen dependent. I also wanted to define here what I'm calling the critical PO2. You'll probably hear me refer to it as P-crit or PO2 crit as we move forward in the presentation. The definition here is just the level of environmental oxygen at which aerobic metabolism cannot happen. So that would essentially look like a respiration rate of 0. And these oxygen conformers exist in contrast to oxygen regulators, whereas these regulators are maintaining their oxygen consumption kind of regardless of the environmental oxygen conditions, only until that PO2 crit is reached. So, this PO2 crit is a really important value for how organisms will respond to hypoxic stress, regardless of whether or not they're a conformer or a regulator. So that brings me to a few of my research questions. The first question I want to outline here is, I wanted to ask, how might acute exposure to varying levels of oxygen affect the respiration and photosynthesis rates of Acropora cervicornis? And what is the PO2 crit for Acropora cervicornis? And then kind of further than that, how will chronic exposure to moderate levels of reduced oxygen affect their bleaching threshold? Will it make them more susceptible to bleaching after they have experienced this kind of stressful condition of low oxygen? And then lastly, are there any genotypes of Acropora cervicornis that are more hypoxia tolerant than others, in the same way that we can kind of determine that there are different species of algal symbiont and different genotypes of coral that are thermotolerant or more thermotolerant than others? And so for today, I'm going to be answering these first two questions, and I hope to address these other two questions in my future experiments going forward. So we set up a preliminary experiment in order to answer that first question of how will photosynthesis and respiration rates be affected by reduced oxygen. We essentially started with 6 different oxygen treatments of different levels of oxygen availability, ranging from ambient oxygen, which is roughly 6 milligrams per liter, all the way down to 0.5, which is considered essentially anoxia. I also want to mention that the generally accepted hypoxia threshold that's reported in the literature is about 2 milligrams per liter. So we wanted to get kind of on both sides of that literature reported value, which is why we chose the values that we chose. Unfortunately, due to limited coral availability, we could only run these tests on 2 fragments of coral, and you'll notice there are also 2 treatments, the 6 and the 4 treatment, that we were only able to run these tests on a single fragment. Certainly going forward, more replication is obviously needed, but again, these are just kind of some preliminary testing results that I wanted to share with you guys today. So after preconditioning for an hour at these different oxygen treatments and at an ambient temperature of 28 Celsius, we then moved them over to these photosynthesis and respiration chambers. So we use this intermittent flow respirometry in order to measure their photosynthesis. And you can see the coral in its nice little chamber over here. So for the photosynthesis measurements, these were conducted under full light so that photosynthesis could obviously occur. And then the respiration measurements were conducted in the dark using an opaque cover over that chamber. So that's how we were able to kind of get that dark respiration measurement. So I wanted to walk you through a little bit of our kind of experimental setup. So here's an image of the setup that we used to collect this photosynthesis and respiration data. First is the pre-sense sensor, which is connected to those chambers that I just showed in the previous slide. It's also connected to this laptop that you're seeing here. And this is how we're able to read the amount of oxygen inside those chambers in real time. Next, we have a solenoid valve, which is connected to a nitrogen tank. And the solenoid valve is controlled by a digital control unit. So this digital control unit essentially opens and closes the valve to allow for bubbling of nitrogen into our hypoxia chamber. And then that digital control unit is connected to a Vernier LabQuest 3 interface. So with this interface, we are able to kind of set the desired oxygen concentration for our hypoxia chamber. And so that's the idea, is that we can kind of set this digital control unit via the LabQuest to our desired amount of oxygen. And the solenoid valve will kick on when it reads a certain value given what we set the digital control unit to. And so again, the idea here is that when we're bubbling nitrogen gas into the water, we're removing oxygen from the water. So that's the way that we're able to kind of lower that amount of oxygen in the water, is just by bubbling nitrogen gas. So here's our actual hypoxia setup. I'm showing the Vernier DO optical probe, which communicates with the LabQuest so that we can see live readings of the oxygen value inside our chamber, which is essentially this bucket apparatus. Next, we have the bubbler that again is connected to that nitrogen tank. Obviously, it's not bubbling right now in this photo, but it would kick on if the digital control unit reads a value that's higher than what we wanted it to be. Next, here are the photosynthesis and respiration chambers, and that chamber would again go inside that bucket when we're ready to take those measurements after that hour of preconditioning. And then finally, I have my 2 coral subjects that are in that bucket undergoing their kind of preconditioning period when this photo was taken. So getting into some of my results, here is a live readout of the Veneer LabQuest data and also the DO optical probe data. And I wanted to share this because I think it's a cool representation of how well we were able to control the amount of oxygen in that bucket or hypoxia chamber. So each colored line represents a different oxygen treatment. I decided to go with a rainbow for ease of color differentiation. On the x-axis, we have the arbitrary time point, and on the y-axis is the amount of oxygen in milligrams per liter. And so you might notice that there are fewer data points in our 6, 4, and 3 milligram treatment. And this is because it took us a little bit of trial and error to figure out what would be the most efficient sampling rate. So to get the water in that oxygen chamber down to 6 milligrams is not too tough. We didn't have to have a very high sampling rate because the communication between the probe and the bubbler didn't need to happen very quickly because the water was already pretty oxygenated. Whereas when we were trying to get our water down to 0.5 milligrams, we had to really increase that sampling rate to once every second so that the probe is continuously every second taking readings and communicating with that bubbler to really push that amount of oxygen down. And I'm sure you can imagine that it's kind of difficult to maintain that level of anoxia in a lab setting. So that's why we kind of increased the resolution there. And it's the reason why we're seeing more data points on that line. So getting into some more of my results, here I'm showing the photosynthesis rates for a single coral that was in all 6 of the oxygen treatments. These rates were standardized to account for the surface area of each fragment, as well as any background photosynthesis that was happening inside that chamber. So I have a graph for each treatment 6 through 0.5 with the oxygen concentration in micromoles per liter on the y-axis and the time point on the x-axis. And I also wanted to point out here that all these graphs are scaled the same way. So the x-axis extends to time point 120, and the y-axis is exactly 50 units for each graph. And I did it this way so that we can kind of easily visually see that these slopes are all very similar. They're all kind of hovering right around 0.4. So we can kind of say from this that there is not really an effect of reduced oxygen on the rate of photosynthesis, which kind of makes sense, right? Because photosynthesis is not really oxygen dependent. So nothing super interesting there. But when we look at our respiration rates, this is where it kind of gets a little bit more interesting. So again, I'm showing these 6 different graphs, one for each of the oxygen treatments. Again, this is the same coral that was in all 6 treatments. These graphs are scaled and standardized as the same way as they were in the previous slide. And here you'll notice that the slope becomes less steep as the oxygen concentration drops. And in our 0.5 milligram treatment, the slope is almost 0. It's negative 0.017. So these results are kind of indicating that respiration did decline under reduced oxygen conditions, which has major implications for how corals will be able to metabolize under stressful hypoxic events in the future. So here I'm showing the combined data of both corals. So this is the photosynthesis and respiration rates for both corals. So essentially each point on this graph represents the slope or the rate of respiration or photosynthesis that I was showing on the previous slide. On the x-axis, we have the oxygen treatment 6 through 0.5. And on the y-axis, we have the slope or the rate of photosynthesis that, again, I was showing on the previous slide. So I was able to do a regression line for both of these plots, and it was determined that reduced oxygen had no significant effect on the rate of photosynthesis. So the slope of this line is not significantly different from 0, given our p equals 0.09. Whereas the respiration rate was significantly affected. So the slope of this line here is significantly different from 0, and we determine that with our p-value of 0.006. So in order to calculate our PO2 crit, the title of my talk today, we wanted to find out where that regression line crosses the x-axis. And so I'm just included a little red line there so you can see kind of where that x-axis is, but this is also known obviously as the x-intercept. This would be where respiration is 0, where there is no respiration happening, which is essentially our PO2 crit. So when we're working with linear equations, it's generally easier to find the y-intercept than it is to find the x-intercept. So to make this calculation a little bit easier, what I did was I plotted the regression slopes against the oxygen treatment, where the respiration rate is now on the x-axis and the treatment of oxygen is on the y-axis. So we've essentially swapped those two axes from the previous slide. And I'm also including this red dotted line to highlight that y-axis. So from here, it's really easy to see where that regression line crosses the y-axis, which is right at roughly 0.3, plus or minus 0.4 milligrams per liter when we include those confidence intervals. And so this little red circle is kind of showing that those range of values that could be PO2 crit. And kind of that area that we're interested in. So, based on these very preliminary results, we can say that the PO2 crit for Acropora cervicornis is roughly 0.3. Even with our error plus or minus 0.4, that's still well below that 2 milligram per liter threshold that's defined in the literature. So from this, we can kind of assume that Acropora cervicornis will survive all but the most severe hypoxic stress, which is good news. And again, certainly more replication is needed to kind of squash those confidence intervals and kind of make this a cleaner line here. So next steps, given that the PO2 crit is so low for acute hypoxic stress, I'm really curious to know how chronic exposure to both moderate and mild oxygen stress might affect a coral survivorship under elevated temperature. So our next larger scale experiment will include preconditioning to 3 different oxygen treatments: our control at ambient, 6 milligrams per liter of oxygen, a treatment that I'm calling mild hypoxic stress at 5 milligrams per liter, and then moderate hypoxic stress at 4 milligrams per liter. And these experiments will also include a much higher sample size, ideally 5 fragments per 10 genotypes. So the idea would be that we would have 50 fragments per treatment here. So after that preconditioning for 1 month, we would then separate our different treatments to expose them to 2 different levels of elevated temperature, 31 degrees Celsius and 32, and then assess their survivorship as days until full mortality, until they unfortunately look like this guy over here on the right. And I also hope to examine any genotypic differences in their survivorship response. And we'll also be collecting tissue data and doing some kind of baseline tissue stuff to see where those differences might lie. So at this point, I have so many people to thank. My committee members: Chris Langdon, Andrew Baker, Diego Learman, and Ian Enox. Very special shout out to John Sebastiani, who helped us set up all the hypoxia equipment. He walked us through all the setup and making sure we had the right equipment. So very special thank you to John. Another special thank you to Allison and Melissa for their coding expertise. There were many a time I was in coding club asking a lot of questions. And then of course my lab mates, Hannah, Hadija, and Yasmin, for their continued support and assistance. And special shout out to Hannah for her photos that you saw in our experimental design. And then of course the entire Learman lab for their coral collection. Without their coral collection, we could not have conducted these experiments. So particularly Martine, Joe, Maddie, and Dalton, but of course everyone in the Learman lab. And then another very special thank you to all of the Langdon lab volunteers. There's too many to list on this slide, but their continuous help with coral husbandry and data collection has been invaluable. And with that, I'm happy to take any questions.
Thank you very much, Emma. All right, questions from our students for Emma.
That was a great talk, Emma. I just have a question for your future directions for I guess you'll be recording until the corals die from the stress, but I was wondering if you were also going to look at like color scores or maybe the reason that they kind of die, because it could be interesting to look for like rapid tissue loss too.
Absolutely. Yeah, that's a really good suggestion. And there, like I mentioned, we'll also be doing kind of tissue analysis and taking tissue samples to see if there are differences in their lipid content and symbiont content. But yeah, the color scale could be a really good way to kind of track track their bleaching response, I guess. And I hadn't really thought of that, but thank you. That's certainly something we can include here for sure.
Anyone from the floor?
Yeah, I have a question on the comment, maybe, Emma, if that's okay.
Yeah, please.
Yeah, so I really enjoyed your presentation. It was very, very clear, and it's interesting data you have. Just in terms of definition, you mentioned that PCRIT, you interpret PCRIT as the oxygen tension where respiration is no longer occurring. And there may be differences among fields, but typically, at least in my field, PCRIT denotes the oxygen tension at which the animal can no longer maintain normal oxygen consumption. So you might want to check on that definition, because it's gonna change your conclusion a little bit, I think. And on that note, you're using a linear equation to regress the relationship between respiration and oxygen tension. And I know that it's a very limited data set in terms of replication, but to me, it actually looked like it's not linear, and that there may be a level of regulation in these fragments, which I think is really interesting. So you might want to look at whether there's a nonlinear fit that better describes your data. And if that's the case, you can actually calculate what's called a regulation index. That'll be a good indicator of how competent the corals are in extracting oxygen at lower oxygen tensions. And then since I have the floor, the last thing is in sort of the field of hypoxia, there's sort of two ways to determine hypoxia tolerance. One is what you've done here, which is depleting oxygen but keeping CO2 constant. And that's fine. You're looking at hypoxia in isolation. The other way of doing it is to let CO2 increase in concert with the decline in oxygen, which is what would happen under normal conditions. So when you have environmental hypoxia, you also have hypercapnia. So I'm wondering if you have any plans to look at sort of the combined stress of reduced oxygen and increased CO2.
Absolutely, yeah. So we have ordered certain mixes of nitrogen gas with a certain level of CO2 in there, so that when we're bubbling, you're essentially getting that nitrogen and that CO2 injection. And we have played around with it a little bit. I didn't share any results on that today, but that's certainly a direction we're looking to go. And I should definitely come and talk to you, and we can discuss all of this more, because it sounds like you'd be an awesome resource. Thank you very much.
I'd love to talk to you about it, because it's really cool what you've done. Just one little note on the CO2 control. One way around it that's really simple is to just close the respirometers and follow the oxygen depletion as CO2 is accumulating. Because in that way you don't have to actually regulate nitrogen and CO2. You're just letting the animal deplete oxygen and accumulate CO2.
Right, absolutely. Yeah, that's a really good thought. Thank you.
Okay, I'm afraid we have to move on. Thank you again, Emma. I'm going to stop recording now.